Hi, welcome to Meet the Experts. I'm David H. Nguyen, and today we're going to talk about a cellular process known as autophagy or autophagy, depending on how you pronounce it. Our special guest today is at the forefront of studying this process called autophagy. He is Qing Zhong. Qing Zhong is an associate professor of internal medicine and biochemistry at the University of Texas Southwestern Medical Center. Qing Zhong studies the biochemical regulation of autophagy in mammalian cells, which includes humans, right? Autophagy is the crucial waste cleaning system for the cell. So when the cell needs to remove damaged organelles, protein aggregates, and invading pathogens, it relies on this process, which we call autophagy. Professor Zhong, welcome to Meet the Experts.
Hi, David. Nice to meet you.
So Professor Zhong and I knew each other from when he was a professor at UC Berkeley. At that time, I was a PhD student, and he was actually one of the people on my dissertation committees and also on my qualifying exam. And I just want to say that because he didn't ask me hard questions during my qualifying exam, today I'm not going to ask him any hard questions either. Okay, though if he wants a hard question, how about let's see. How can we tell the difference between when a membrane, the curved membrane unfolds, how much is it due to the forces that cause the curvature and how much is it due to the thermodynamics of just membranes, lipids unfolding? That was a joke, by the way. I just threw that out of nowhere just for
That's a hard question.
Okay. Well, we'll get back to that at the end of the show. Okay. All right. Well, right. So let's get into the questions and answer part here. Okay. So the Nobel Prize in Physiology or Medicine for 2016, as last year, was awarded to Yoshinori Osumu because he discovered the process of autophagy decades ago. Now, my question to you, Dr. Zhong, is there a mixture of disappointment and excitement that your area of research won the Nobel Prize, but that this is now all the harder for you to win the Nobel Prize in the same field?
Well, I think all the scientists in this field are very excited to hear the great news that Yoshinori Osumu win the Nobel Prize. I have to say that many scientists deserve to be at least on the short list of this prize, but definitely Osumu is the one in everyone's list. He has done such fundamental, important work, great-breaking, just really opened the field for all of us. But several scientists such as Beth Levine in my institute, Dan Kriolsky at the University of Michigan, and Noboru Mitsushima at the University of Tokyo, Tomatsu Yoshimori at Osaka University, and Michael Sam at the University Medical Center Göttingen at Germany. They all deserve to be mentioned for their contribution in this field. It's a pity they didn't have this type of recognition, but I think the field will also appreciate their contribution as well.
And do you any discussion or thoughts as to why only he received it and no like two other people, right? Just him, right?
Yeah, that's kind of surprise to all of us. Originally, we think it should be three of them. When we guess who is going to get the award, we make all the different combinations. Maybe he should win the prize with his two students. Maybe he should win the prize with two of the East scientists. But I guess he's the only one without any controversy because everybody will put him on the first of their list. This is why he got the prize.
Well, I'm sure there's other prizes that people in your field, including yourself, can win. You know, Howard Hughes, National Academy of Science. So, there's still a lot to be.
That's true. But the field is still young, and we have many opportunities. Yes, definitely. Yes.
Yeah, and so definitely you publish in the top journals, right? Nature, one of the top journals out there. So definitely you're at the cutting edge, in your field. So that's something that should be highlighted.
Yeah, thank you, definitely. Yeah, well, we are not really just pay so much attention to the Nobel Prize by itself. Nobel Prize gives to the most original work, But we are more focused on the question itself and really the potential implication behind the question, such as how it's being related to human diseases and what kind of maybe the therapy breakthrough can bring to the different diseases. So yeah, we pay more attention to these questions.
Okay, well, that segues perfectly into our question. That is, how does autophagy work?
Well, yeah, that's a amazing thing, because in 1960s, when people have this kind of electron microscope, people think they already know everything about the cell, but actually it's not. They observe this kind of very unique membrane vesicles. It's just like a bag. They can put many big content, big items in the bag, such as intact mitochondria and intact ribosome ER and all these different organelles into these membrane vesicles. Those they don't really know how to call it. So I guess the first name is from a Greek word called autophagy, means self-eating. I guess people have no idea what it is. The initial definition of this autophagosome, which is self-eating vesicle, is only described as membrane-lined vesicle with cytosolic content inside. So we can think how simple that definition is. So we have no clue what it is. But later on, so people realize this is a recycling system. They put all these damaged organelle into this membrane vesicle and deliver them to a lysosome for degradation and recycle. That's become very important because all the human bodies is composed of the minimum functional unit, which is a cell, right? So we have cells in different organs. In order to keep a cell healthy, so we have to recycle our organelles from time to time. Just like a car, so one car can run like 10 years, sometimes a car can run 50 years. It really depends on how you maintain them. So if you can recycle your transmission and all this air conditioner very frequently, you can keep it as a new car, even after use for 50 years. I guess this is the principle for the autophagy. They can keep all human cells healthy in very relatively long time. So you don't have all these kind of toxin eventually will kill our cell and cause all different types of diseases.
Okay. And so, and it sounds like you touched upon this a bit, but are there ways in which autophagy is activated in cells?
Yeah, the initial clue comes from the starvation. Okay, so when no matter it's human cell or yeast cell, when they face nutrient starvation, they don't have food. So the first response is not to kill themselves. Actually, they're trying to engulf the cytosol or the mitochondria or the organelle in the autophagosome and deliver them to lysosome. So they can break into amino acid and fatty acid, or these kind of all building blocks for the urgent use, such as they can bypass all these kind of crises. So they can live for a relatively long time. That's why a yeast, when you starve them, and a human cell, when you starve them, they don't die immediately. They can use this kind of mechanism to survive for quite some long time, such as human cells can survive up to 2 weeks. But if you don't have this autophagy system, they will drop dead. So they cannot tolerate any harsh conditions. So that will be a disaster for all different kinds of living organisms.
And so then, as a side question, so is autophagy related to the life extension due to caloric restriction, right? Eating less, living longer. Is this part of autophagy? Do you think it's part of that?
Yeah, definitely. That's really the very exciting hypothesis in the field. So by recycling our damaged organelle and long-lived protein from time to time, we can renew our cells in terms of organelles and in terms of all different kinds of proteins. Actually, we can prolong our lifespan. The direct evidence is no matter what kind of method you use to prolong our lifespan, no matter in yeast or in C. elegans or in the human or in mice, so they all require autophagy to sustain this kind of lifespan stimulating activities. So there is a very exciting hypothesis saying that autophagy activation might be required for lifespan extension. And some drug can stimulate lifespan extension, such as metformin. They also stimulate autophagy. So autophagy definitely play a role there. But we just don't really know at this moment how big the contribution autophagy has in the whole process. It has a role for sure.
Mm. All right. So, what are there any human diseases that result when autophagy?
Yes, definitely. I think the first one is people ask is cancer. Do we need autophagy either for the tumorigenesis or to prevent cancer? That's been controversial in the field. Originally, people think if you don't have autophagy system, the mitochondria cannot be healthy, so they will generate ROS, eventually will lead to cancer. But later on, people realized that tumor cell actually can utilize, or you can say hijack this autophagy system. They can eliminate the chemotherapy drug as a toxin. So they can reduce the toxicity caused by chemotherapy. Actually, tumor cell can use this one as a survival system to bypass all these nutrient shortage they are facing when they do this kind of very aggressive growth. So in that case, autophagy is actually helping tumor to grow. So if we already have a tumor, we want to shut down the autophagy program to suppress the tumor cell growth rather than promoting it. So I'm just saying they have these two sides functions. Yeah, we probably normal cell we need autophagy to maintain. Otherwise, we can shut down the autophagy to prevent tumor cell growth. Autophagy also have a very clear role in neurodegeneration. As I mentioned, it recycles organelles. Some of the neuron cells can survive up to 50 years in our body. So if we don't recycle the damaged organelle, those cells will eventually die. If we can stimulate autophagy in these cells, they can live longer. So this is why without autophagy, neurodegeneration happens way earlier than it should be. And also about this bacterial infection, because most of them, or a lot of them, are eliminated by the autophagy system to be degraded in the lysosome. And insulin response and diabetes is also regulated by autophagy. If you look at the literature, essentially you can find all the major diseases are somehow regulated by autophagy. So it's been involved in a broad spectrum of human diseases.
Wow, that's amazing. So here's a tangential question as you're talking about cancer and autophagy. In the cases of when two cancer cells fuse, cell fusion, right? The fused cell has two nuclei, but do they get rid of certain organelles, and is that autophagy or no?
That's actually a very, very interesting question, which is I don't really personally know, because people have to design the proper experiment to answer that question. But many of our systems actually use this autophagy to eliminate their organelles. One of the great examples is red blood cell. Our red blood cell, they don't have nuclei. So that is eliminated by autophagy. That is already known.
Wow, all right. And so have there been any therapies specifically targeting the autophagy pathway in treating diseases?
Yeah, that's a very question. Currently, there's no specific drug. This is why I mentioned there's many things to do for our biochemists. This is our really the future goal. So by understanding the biochemical mechanism of autophagy, which is we know exactly which enzyme is the most important enzyme in this process, maybe you can target this key enzyme and produce or design the drug that can specifically target autophagy. But currently, there's many kind of crosstalks between drugs and autophagy, such as many drugs, they have the activity, they can stimulate autophagy, such as metformin, by treating diabetes, they can stimulate autophagy, such as chloroquine, which is originally used to treat malaria. So now they are being used to block autophagy because they can suppress the lysosome activity, which block the degradation of the autophagosome.
Well, definitely. So a follow-up question would be then, what are current challenges and the future frontiers of studying autophagy?
Well, there's many different aspects of autophagy we don't really know, such as many very fundamental questions. The field, the scientists in these fields are very interested in such questions, such as where is the membrane of autophagosome originally come from? In our hair cells, essentially all the membrane, they're budding from somewhere. They don't really generate by itself. So all the membrane are essentially budding from ER and maybe plasma membrane. But autophagosome is a mysterious thing because we don't really know how the autophagosome membrane is originated from. We have all kind of speculation, and ER is most likely the source of autophagosome membrane. But even it's probably going through a very different, unique way to form the autophagosome, which is something we still don't understand at this moment. And the second one we want to ask is, we already know there are so many players in the autophagy pathway. We have probably 40 autophagy genes. That's more in the mammalian cells, but we don't really know exactly how they make this process work. So this is actually my personal interest, and many other scientists working on this aspect as well. So we need to know exactly which protein function in which step to make this process happen. Only by knowing these details, we can design drugs to target individual steps, to block or to activate the whole process, and hopefully to treat different diseases.
Wow. And so you said earlier that the membrane, the source of the membrane in the autophagosome is different than that of the ER. Is that right? And so it's a mystery as to where the lipids in the phagosome. Is that?
Yeah, definitely. I think at an early time, people are very confused. Most of the membrane, when they're coming from ER, they are protein coded, which is like they're very close. So these close make this membrane budding from the original membrane. But when people look at the EM of the phagosome, they are not really happy about that because they don't see anything. The older autophagosome is almost naked, so they have no clue what membrane coating on the membrane. So it seems like autophagosome is formed through a very different mechanism, not the budding mechanism. The second, autophagosome is very unique in a way. They have double membrane. Most of the membrane we have is single membrane. It's a dual layer, but it's a single membrane, such as plasma membrane, such as all these different organelles membrane. Mitochondria is different, it's double membrane. But autophagosome is another very unique thing. It's a double membrane. So nobody understands at this moment why they form double membrane and how the double membrane is formed. Anything inside the double membrane, why they need two membranes instead of one. So they're all open questions.
Yeah, fascinating. And so in your part of your lab, one thing you study, within these membranes is this topic of membrane curvature. Now, can you describe what that phrase means and why membrane curvature is important?
Yeah, membrane curvature, well, that's because membrane is so difficult to study. So we all know that membrane is formed by lipids, right? Lipids has its orientation. So they have these polar ends pointing outwards. They have these non-polar ends pointing inwards. So you can imagine the surface of the lipid or the membrane is so slippery. Nothing can attach to it. A protein, if they want to associate with membrane, is extremely difficult. So sometimes they can generate a specific type of the lipid, which is phospholipid. And protein combines to the phosphor group of the lipid, they can attach to the membrane. Another very interesting mechanism is membrane sometimes they form curvature, which means they form different shapes, right? If it's a tip structure, the curvature is very high. So membrane curvature, why is different from a curved membrane? Why is different from the flat membrane? Because they expose more space in between. So they expose their non-polar side, which is hydrophobic. A protein can insert into this exposed space that can bind to the curved membrane. So membrane curvature sometimes become a very, very attractive place for protein to bind to. It's essentially the starting protein to bind to. So we always think membrane curvature recruits the first protein to bind to the membrane, and this protein then bring the rest of the protein to the membrane, then the whole complex will eventually cover the membrane. So this is the so-called sequential events of this kind of protein recruitment.
Okay, wow, yeah. All right, and so if you're just joining us now, we are interviewing Professor Qing Zhong, associate professor at the University of Texas, Southwestern Medical Center. And his first name is pronounced Qing, but it's spelled Qing. So it's one of those things where it gets a lot of people probably, right? They say are you Qing or Qing, but it's Qing,
yes.
Right? So Professor Zhong, can you? Most of our audience members are high school students, college students, parents, and also college counselors, right? And also a lot of practicing scientists out there who are interested in cutting edge science. So could you describe your academic journey starting from high school?
Well, I have to say I don't really have much science exposure in high school. As any student in China, our high school is very busy in terms of preparing all this entry exam for the college. When I'm trying to figure out which university I want to go to, I talked to my parents. My parents used to ask me, what do you want to study in college? I said, I have no idea. That's the only thing I know about. I want to study something about human. Anything human related will be good. I don't want to work on machines, languages, and all these different things. I want to work on humans. So in that case, medical school is a no-brainer choice. So no-brainer. So I'm fortunate to get in this top medical school in China. So they have this kind of 8-year program. In the first 2.5 years, we study very basic biology, and next 1.5 year we study the basic medicine, and then we go to the hospital and do this kind of residency. Yeah, it's very interesting actually. Although I figure out I'm not really good at memorizing things, but I still appreciate all this medical school training. But my real interest really comes late. I remember when I was in my 8th year, which is I'm already pretty old, we have this kind of internship for science. So it's an 8 months science program. At that time, I joined the lab. I studied spermatogenesis, which gene control the sperm and how they develop. And it's quite interesting stuff there. I'm trying to purify 2 transmembrane protein, which is very, very difficult to purify. I have no idea at that moment. I'm just saying that I really work day and night. I can't figure out anything. At the end, I have to say I failed the program, but I really being activate or just my curiosity is booming up after this kind of practice. I really want to know what's going on there and how we can learn anything from them. Is there any way I can figure it out? And after that, I graduate. Actually, I joined the hospital, be a doctor for oncologist for 1 year. Again, I like to be a doctor at the first place, but I eventually figure out my curiosity just can't be satisfied. I want to know more. I want to know what is behind this kind of cancer drug and why this one has this side effect, how we can improve and stuff. So I decided to apply grad school in the States, and hopefully I can study some kind of basic mechanism of cancer. So I applied. I have 2 offers. One is LA, and the other one is San Antonio. I was trying to decide which city I should go to. It's LA or San Antonio. I figured out that LA has too much temptation there. If I go there, I probably can't resist. So I decided to go San Antonio. I heard that it's nowhere, just you're sitting desert, and that's the only thing you can do, just work in the lab, I guess. So I joined the lab working on tumor suppressor genes of breast cancer. Yeah, it's pretty exciting 5 years. We figure out something, we publish good journals, and then I decide where to go for the postdoc. I joined UT Southwestern because there is a faculty here really good at using the biochemical approach to figure out a pathway rather than individual genes. This is why I really want to, I really have this kind of problem with myself, just as when I'm doing my graduate study, although I'm doing okay, but I just don't like the idea. I always start from one gene and trying to connect this gene to different aspects of biology. Sometimes I really want to see, maybe we should start with a pathway so we can decide within this pathway which is more important, so we can concentrate on this most important thing and how it's being related to the cancer to save our time. Anyway, I did that in my postdoc area. We studied apoptosis, which is a cell death pathway. And I did okay. Again, I published the journal, and I had this opportunity to apply a faculty job somewhere. Then I decide I want to change again because I'm not really satisfied with studying a pathway for my rest of my life. I want to working on a new pathway, which is totally new, so I can really contribute to this field, which is autophagy. In the time I started my lab at UC Berkeley about autophagy, there's not much known about autophagy, especially the autophagy function in the mammalian cells. They have a few genes identified, many of them has not been identified. So I participate, I'm the one of the groups to identify AG14, which is the crucial gene in autophagy, and along with several other labs. So it's become very exciting to us. Our dream is to, maybe you can figure out, we want to put different parts of autophagy together. So we want to see if you can assemble the whole thing into a car so it can run by itself to do the autophagy job. But now I figure out that's probably too difficult for us to finish. Now we are trying to figure out different parts, such as maybe you can assemble a tire first, or maybe you can assemble an engine first, which is tricky as well. So now we are looking at different parts of autophagy, trying to see if we can reconstitute by different proteins in a test tube. I guess now I get tenured. I'm trying to try something new again because we are not really satisfied just testing all this in my test tube. And now we are trying to see the animal phenotype. If you claim your gene is important, can you really prove it in the animal setting? So will that be something like humans? They develop different diseases, or can you set up a platform for the future to screen drugs and to really solve some problem.
Wow, okay. That's very inspiring, your journey. And one thing I want to highlight for our student audience is that earlier you said you did 8 months of research and you felt you worked day and night and you couldn't purify these membrane parts that you wanted to, and you felt like you failed those 8 months, but really that failure was what pushed you to be more curious, right? Or to maybe
Yeah, I have to say that. That's quite a challenge for me. I'm just the people, I like challenge. I'm very curious. I want to know what's going on there. If I can't get it, I just don't want to stop. I really want to figure out what's going on. I guess that's why I trapped in this science field at the beginning.
That's funny that you described that you're trapped in the science field.
Yeah. But later on, many people mentioned to me, "Oh, okay, you should go get the board exam and you should be a doctor in the States." I have to say, I almost forgot I had this opportunity. I was too busy working in the lab trying to fight with my puzzles, and sometimes I call it a game. I'm really trying to figure out what's going on there and see what's the best strategy and how to solve the problem. But ultimately, I'm still interested. I'm concentrating on one single question, which is, I guess, very difficult, just how to treat cancer. So I really try and find if there's a new way or new angle to look at cancer, to find the weak spot, right, of the cancer, and maybe different cancer have different weak spot, and maybe you can target it and find a way to cure them. I'm not saying, what you see the tumor immunology and now become very popular because not 10 years ago or 20 years ago, nobody think that that will be a huge breakthrough, but now it's become overwhelming. I guess my field and many unknown field might have the similar function or possibility in treating cancers in the future. Who knows? Let's keep our eye open, and there might be something in this field happening as well.
Definitely, definitely. So, and here's a question that we ask each of our webinar speakers. So it's easy for students to be inspired by someone like you, but it's also easy for them to be intimidated by someone like you, right? Who was successful in high school and college, med school and PhD, right? And now you're tenured professor. And so, were there times in your academic journey when you felt inadequate and that you wanted to quit?
That's an interesting question. I guess there is many. We have many frustrations, right? So same thing. I have many frustrations. When I first get into graduate school, I was so frightened. So I almost failed my first exam. So I was told if I didn't pass my exam, I will turn into a master's degree rather than a PhD. I cannot continue. Yeah, I was thinking about that time. Maybe I should change my career. But again, as I mentioned, just sometimes I guess you need some kind of discipline. So if you know what you really want to do, so you have to make a plan. You have to figure out what's the best that you can do, and just go ahead and do it. Don't think too much. I guess this is very important. Just don't think too much. Go straight for your aims and goals, and then you will find it's quite stimulating afterwards. I always tell my students and also myself, now I really enjoy to be a scientist. Because why? Because I can't imagine myself to be a doctor, because I think it's so much pressure to be a doctor. You have to be perfect in many different ways. When you treat a different patient, how can you fail, right? They're all patients, they all just begging for the best choice of their life. But to be a scientist, you have the luxury to face many failures. So I can fail 1,000-time experiment. If I figure out the right condition, if anything worked, so I can have this perfect result of this discovery nobody made before. I really enjoy this feeling, and eventually it's kind of fulfilling, really make me happy. And yeah, it's not really about money, it's not really about positions, it's all about this kind of discovery and all this kind of achievement just finally make me here. Yeah.
All right. And so what advice do you have for current high school students and college students who want to pursue a career in your field?
I think the most important thing, I can't speak for the most of scientists because I don't really think I can representing them in any way. But to myself, I think the most important thing is keep your curiosity. I'm so lucky to keep my personal curiosity. I never feel this is something miserable or something I don't like, because I'm always curious about what I'm doing. I really want to know the answer. And I think at the very beginning, you have many frustration because you don't really know how to do it. Then the second thing comes in, you need to have discipline. You need to make plans. You need to figure out, learn from the best, and do the most reasonable way, and achieve the goal before you give up. So I'm just saying it. And the third thing, I really want to students and my colleagues. Yeah, the third thing is just appreciation. So you have to appreciate yourself's effort. You have to award yourself from time to time. And you have to appreciate the help from others. You have to appreciate help from all over your surroundings and the environment. And because all these possibilities, so you can make progress. with this appreciation, then you can move forward without too much regression. So I'm just saying, yeah, if you keep your curiosity, you're doing something you like about, you are just keep your appreciation, and I think you will be a happy guy and doing whatever you like.
All right. And so what type of background courses does a student need to do or take in order to do the research that you do?
Yeah, for sure, biology. You should know biology. You should know this kind of history and evolution and biology. Personally, I really regret. I should be very serious about chemistry because I really want to learn. If I have chance again, I want to go through the college chemistry again. Chemistry is very, very important. Now, another thing become very, very important as well, which is computer science. So if you know, because we have so many data, because of all these kind of human genome project, because of all these different kind of studies, we have huge amount of data. So if you know how to do the programming and computer science, if you know chemistry, physics, and biology, you can become a great scientist. You know, you can have the great vision to look at science in a different aspect. I think the biology still is the brightest area in this century because we are so curious about human beings. Originally, I think in the past 20 years, we can probably only working on the monogene disease. Now, scientists are really trying to figure out what's going on with these kind of complex diseases, such as all different cancers, diabetes, obesity, and neurodegeneration. We have, well, I can't say we are successful in this aspect, but just like Edison said, we already tried 1,000 methods, we all failed. So, which means maybe the next generation, they have the great opportunity to success in any of these aspects if they have a unique vision or you have a new technology. I'm just saying that would be a fantastic era just to look at the science in a totally different ways if you have enough background and knowledge.
Wonderful. All right, and so that's the end of the Q&A with me. Now turn you over to our marketing director, Alistair.
Hey everybody, I am your moderator for today, and we do have a couple of questions. I want to thank Dr. Zhong for sharing his expertise with us today. First question is from Jen. Jen says, I am very interested in nutrition and biochemical sciences. So she asks, how long do you actually need to fast before the activation of autophagy actually activates?
Yeah, that's original people think that's take a couple hours, but now we all know that if you have amino acid starvation, it only takes 30 minutes. Could be even shorter. So after 10 minutes, we already see that. The nutrients have a very important relationship with autophagy, not only just amino acid. Now we think maybe the metals can induce autophagy as well, such as zinc. If we deplete zinc, in our body or in yeast or in different organisms, so they can induce autophagy as well. I'm just saying the nutrition deficiency it's a very strong trigger for autophagy. So yeah, it's very interesting to figure out what's the mechanism here. It's very rapid. Yeah.
So I guess this might be my own personal question, but there's some diets out there, and a lot of them actually recommend intermittent fasting, and part of that diet means that you kind of do a fast for about 14 hours a day. And at what point do people have scientific evidence of at what point fasting actually becomes unbeneficial? It actually hurts you before you actually get benefits from it, from the autophagy.
Yeah. First thing, the calorie restriction is so far the only way can prolong lifespans. So this is being scientifically proven, that's for sure. But they do have a limit. You cannot reach too far, you cannot do too long, such as when we do the diet restriction, normally we ask them to have 40% of normal diet. So which means you probably can't go less. 40% is good enough. And I guess you probably can't fasting for too long as well. I think at the most you can fasting about 1/3 of your time, and you can go back to normal diet again, and such as 2 days in a week, and you will go back to normal diet. But I guess you cannot go too long beyond that because all we want to see is we want to stimulate autophagy. At the same time, we want the damage to be reversible. So you need some time for the cell to recover, and you cannot damage them to a limit so that they cannot recover. So this is very important to keep your limit in the right range.
Okay, this next question is from a student. His name is Ben. His questions ask how you described autophagy and your research about using it against cancer. Sounds like it's the road to the fountain of youth. Can autophagy actually be used for extending life and your youthfulness? If so, how far are we actually able from achieving that?
Yeah, so there's always dilemma talking about aging and the cancers. Many early attempts, they're trying to prolong lives, ends up getting cancers. So I guess the cancer cell is most like our normal cells. They love autophagy as much as a normal cell does. So they all love autophagy. They're trying to take advantage of autophagy system to survive themselves. I guess there must be a limit how to do that. I guess the current dogma is if you have no cancer, you're perfectly normal. Autophagy actually is doing a good job. They are recycling the organelles. You expect to live longer with active autophagy, which has been already more or less supported by the animal experiments. But at the same time, when you already have cancer, then probably this is not a great idea because autophagy stimulation may help the cancer cell to get by all these therapies. In that sense, autophagy inhibition might be a good strategy. But again, I guess it's too early to say how to balance these two, autophagy stimulation, autophagy and inhibition, unless maybe someday we can find a drug we can test in the animal so we can really know what's going on there. So I guess it's too early to say at this moment. Currently, we have no specific drug targeting autophagy yet. Make this question very difficult to address at this moment.
Okay, the next question is from Sean. Sean asks, tell me more about the gene that you identified to be directly linked to autophagy. Have you guys actually tested that gene and tried disrupting it to see what it would actually happen to a living organism? Or is that something that you guys are working on at the moment?
Okay, so when we first started our lab, so we have this question. So in the autophagosome, there is a lipid called PI3P. PI3P is so important because if you have the PI3P, they can recruit protein to the membrane to initiate the whole process. But at that moment, we don't really know how to recruit these kinases to the membrane because those kinases cannot bind to the autophagosome membrane. So we need some protein to recognize the autophagosome membrane and recruit the complex to the membrane. That one is missing at that moment, so we're trying to identify this protein. So we demonstrate, along with other groups, we demonstrate the protein we identified called ATG14 is indeed this type of protein. As David just mentioned, ATG14 will recognize the curved membrane on the autophagosome membrane and insert itself into the membrane. So this is the first protein to be on the membrane. With this protein anchored to the membrane, it can recruit the rest of the protein come to the membrane and to initiate the whole process. So we think that will be the first specific protein to targeting this process. Now we think it's very important because we are trying to target the whole set of these lipid kinase using for drug discovery. So what we are going to do is we are going to have all these proteins purified, put them together in a complex, and through compound into this reaction to see which compound can specifically target this lipid kinase. Hopefully, by doing this way, we can figure out a compound that specifically target autophagy and might have a function in cancer treatment as well.
Okay. That's it. That's all the questions we have. Thank you, Dr. Zhong, for spending your afternoon with us.
Thank you.
Yeah, I just want to thank you, Professor Zhong, for being here and sharing your perspective on your research and your educational journey. I'm sure of our students who are also of Chinese descent, will enjoy, hearing your perspective. And we look forward to having you back on the show to, I guess, interact with different panels on different.
Yeah.
Thank you very much.
No problem at all. Thank you for having me. Yeah, it's a great pleasure. Yeah.
Thank you.
Thank you.